Please join me in welcoming to the Distinctive Voices podium, Dr. John Evans.
Great, thank you very much. It's quite an honor to be here. I want to start with some truth in advertising, which is that I am not a scientist. I am not an ethicist. I am a sociologist, the worst type. I actually don't make ethical claims in any of my work, but I've spent the last 20 years studying ethical debates by both what we would call elites and the masses. And so I'm going to describe this debate for you that I thought had kind of gone away and I'd kind of moved on to other things, but technology has brought it back to the fore. So, what is gene editing about? Well, it's about human genetics. This is DNA. Every cell in your body has genes in it. There are chemicals. These are chemicals that determine what your cells do. Humans normally have 23 chromosomes, each of which contain genes. There are thought to be 20 to 25,000 genes in the human genome. They're arranged like this, as Crick and Watson first showed us in 1953. The particular genes that you have results in the qualities of all the cells in your body. Therefore, some of us are tall, some are short. This is at least partially caused by your genes. Complex traits, like intelligence, are thought to have a genetic component. Intelligence is not due to one gene, but at best, a large number of genes that interact in ways that no one understands. A lot of diseases are genetic. We're familiar with the ones where only one gene has gone wrong. This one gene is typically the linchpin for something your body really needs. So, for example, sickle cell anemia, cystic fibrosis, Huntington's disease. In sickle cell, one little mistake in the gene results in your blood cells being the wrong shape, which causes all sorts of health problems. Particular gene combinations make you susceptible to disease, but that's not very well understood. And as we know, you pass your genes on to your children. So, that's it for your science lesson for today. That's the extent of my knowledge. And when it comes to the technology here, I just rely upon my colleagues to tell me what is possible and what's not possible. And I also want to do it this way to let you know that you do not have to have a deep understanding of the science to have an opinion about the moral stance towards these technologies. So, if I were to change your genes, I could change you. Let's take an example of what scientists have been trying to do for years. If you remember, this is John Travolta. I see some young people up in the corner who might not know who that is, but there is something called boy in the bubble disease, actually known as severe combined immunodeficiency, unbelievably rare. Because of a genetic defect, one gene wrong, you essentially don't have an immune system and you have to live in a bubble so you don't catch any diseases. One of the first attempts at changing the genes inside of an existing person was in the early 90s. How would you actually do that? Well, the first problem is you have to get at the cells that you want to change. So you can't get at the cells on the inside of your eyeball. You can't really get at the inside the cells in the middle of your brain, but the cells that make your immune cells are in your bone marrow. You can take them out, you can manipulate them, you can put them back. They've been trying to do this for years for all sorts of genetic diseases. But how would you do that? This is for a different disease, but it's the same process. Basically, what people have tried to do is get the DNA you need, put it on a virus, and let that virus infect the cells that you want to change. Okay? Couple problems with this is that it doesn't land at the right place in the genome. It can sort of miss and then cause problems from that. It might only infect 8% of all the cells that you want to hit. Okay? This virus itself that delivers this can cause problems. Someone died in a gene therapy trial in 1999 simply from an immune reaction to the virus. A 2002 trial in France with these SCID children stopped after they developed leukemia because the theory is that the virus sort of landed in the wrong place. The gene was delivered to the wrong place and caused leukemia. I think the summary here would be that this doesn't really work very well at present, and that's why people have only tried it on the most extreme of diseases, like where you'd have to live the rest of your life in a bubble. But what if you could really accurately put this exactly where you want it? In every single cell you tried to do this. There were no off-target effects, super high accuracy. Well, a few years ago, a major improvement was made. So this thing here at the top stands for, if you pause here, clustered, regularly interspaced, short, palindromic repeats. I will not say that ever again. We will summarize that with CRISPR, okay? You do not need to know how this works, okay? You just need to know that the people who do understand how this works say it works incredibly well. It's very, very accurate. So accurate, indeed, that the metaphor they're using is gene editing, as in your word processing program, you could tell your word processor, go look for every three-letter sequence like this, and when you see it, replace it with this other thing, okay? So, if you actually know exactly where the gene for boy in the bubble disease is, you can search it out, replace it with apparently incredible accuracy. The tools are now in hand to heal boy in the bubble disease and all sorts of other things like that they've been trying to do for decades. This is not ethically controversial. Most people consider this to be sort of medicine. Now, the ethical challenge becomes, you can use CRISPR for any cell, including a human embryo. What immediately became controversial is some Chinese scientists who decided to use CRISPR to modify a human embryo. They did it with human embryos that weren't viable to begin with. They had no intention of implanting this in a woman to make a baby, but it was a proof of concept. It didn't work, but scientists tell me that it could work. And indeed, a few weeks ago, the British government approved the scientists there attempting to do this without putting it in a woman when they're done. So, what this would mean would be, you change an embryo and you put that in a woman. But the key here to remember is that that change would make a changed baby, but then it would change that baby's babies, that baby's babies, and so on down the line. So it's essentially eventually changing the species. So something very rare happened. Scientists themselves, including the ones who invented this technology to begin with, called for a moratorium on modifying human embryos that will be put into women and allowed to grow into babies. No CRISPR babies. They published this consensus statement here in Science magazine. They were primarily concerned about safety, as no one really has any idea what exactly would happen to a resulting baby at this point. Just to give you one example, if you do this on an 8-celled embryo and you don't get to all the cells, you have what's called mosaicism. What if 7/8 of your cell have one genetic sequence and 1/8 have another? So, open question here. Very quickly afterwards, scientists held this international summit under the auspices of the National Academy of Sciences, the Royal Society, which is the British equivalent, and the Chinese Academy of Sciences. This is the chair of that summit, David Baltimore, who was one of the scientists in the 1970s and 80s who figured out how genetic engineering in general would work with microbes and the like. He was also one of the organizers of the last time scientists called for a temporary moratorium in the name of safety, which was at the Asilomar Conference in 1975, which was concerned with genetic modification of things like E. coli bacterium. And the fear then was, if you modify one of these things and it escapes from the lab, would it wipe out all the regular E. coli, and what would that do, et cetera, et cetera. So there's been all sorts of magazine, newspaper articles about this issue. This is from The Atlantic. What's the fuss? So this talk is about the fuss. But to anticipate where we're going, in this Atlantic article about CRISPR, one of the inventors of the technique was interviewed. And as background, you should know that scientists who become involved with public debates about these sorts of technologies generally try to avoid making statements about the social implications of these things and try to keep discussions in the realm of safety and other areas for which they have expertise. But that was not in this case. In the article, one of the inventors of CRISPR told the reporter she has this dream. She says, in my dream, this scientific researcher had come to me and said, I have someone very powerful with me who I'd like you to meet, and I want you to explain to him how this technology functions. So I said, sure, who is it? It was Adolf Hitler. I was really horrified, but I went into the room, and there was Hitler. He had a pig face, and I could only see him from behind, and he was taking notes, and he said, I want to understand the uses and implications of this amazing technology. I woke up in a cold sweat, and that dream has haunted me from that day, because suppose someone like Hitler had access to this, we can only imagine the kind of horrible uses he could put it to. So what is she referring to here? This is not how scientists usually like to talk in public. To get further into this, we have to dig deeper into the history of these technologies and debates about genetic modification. So, this is about as complicated a table as I will get. So, here's some ethical distinctions that were really set in place in the mid-70s. In the ethical debates, we could think about 4 categories. The first divide is between somatic and germline, which I explained a minute ago. A somatic change, soma, the body, means I could change a gene in one of you, okay, but that change would die with you. It wouldn't be passed on to your offspring, okay? This is generally considered to be less ethically fraught because it just is with you. Germline means I change you or an embryo or a sperm or something, but that it gets passed on to children, those children's children, and so on, affecting the germ line. The other divide is between diseases and enhancements. So, trying to use genetic modification to heal diseases has been much less controversial than enhancement, such as creating people who are more intelligent than others, for example. So the whole Hitler reference sits in cell 4. So if we look at the history of this, we have to start with the late 19th and early 20th century eugenics movement. Eugenics means improving the genetic qualities of the human population. The idea here was that the genetically better, quote-unquote, people should mate and have more children than the inferior people. If more of the better people have more babies and the inferior people have fewer babies, eventually society would improve genetically. At least that was the idea. This is America's most influential eugenicist. He wrote in 1911 that the genes of Poles made them, quote, independent and self-reliant, though clannish. Italians tended towards crimes of personal violence, and that the Hebrews were predisposed to thieving, though rarely to personal violence. The peak of these racial eugenic ideas in America was 1924, when the Immigration Control Act was designed to limit people from Eastern and Southern European, keep them from entering the United States. And it was signed by President Coolidge, who had earlier said, quote, America must be kept American because biological laws show that Nordics deteriorate when mixed with other races. So this was not an uncommon notion in the early part of the 20th century. And this is one way to control the germline of the population, at least in a country, is you stop entire groups of people from showing up here to intermarry with everybody else. I don't know if you can read that. But in the 20s, these ideas started to become separated from race, and geneticists became concerned with good and bad gene people of any race. This is from the Kansas State Fair. There was always these movements to try to get people to only have fit marriages and avoid non-fit marriages. Note the human traits here that were thought to be genetically determined, such as feeble-mindedness, which is a classic, epilepsy, criminality, pauperism, and others, okay? There was, contests where you could have the most fit family, you'd win a big thing here on your, there was sermons for the, most fit family, eugenics sermons and the like. Now, this was not all voluntary. Many people were forcibly sterilized in the name of stopping the degeneration of the species. This is Carrie Buck with her mother. Carrie was famous for an infamous 1927 Supreme Court case that involved her forced sterilization because she was a quote-unquote moral imbecile. Another supposed genetic trait that people at the time thought was passed down. Justice Oliver Wendell Holmes of the Supreme Court wrote the opinion that allowed this and concluded that sterilization was justified because, quote, 3 generations of imbeciles are enough. Today, we would presume that whatever behavior is being referred to as moral imbecile, it is learned behavior, not genetic. Eugenics of the era was actually a progressive social reform thought to be a way to stop poverty and help the people themselves. California was actually the national leader in coercive eugenics, forcibly sterilizing the most people in the United States. Of course, the eugenics movement also existed in Europe, very strongly in Germany. Nazis took this to an extreme. This is a poster emphasizing the cost of feeding one person with genetic disease versus an entire family of fit Germans. Early on, the Nazis actually learned from California policies and practices, eventually passing a law, a sterilization law in 1933, similar to what was being done in California. Later on, Germany did not just sterilize the quote-unquote genetic defectives, but it killed them. And the idea of eugenics culminated in the Nazis' conclusion that entire races or ethnicities were genetically impure and needed to be eradicated, most notably Jews and Roma in the Holocaust. According to historian Daniel Kevles, after the revelation of the Holocaust, people realized that, quote, a river of blood would eventually run from the German sterilization of law of 1933 to Auschwitz and Buchenwald. Well, you might think this was the end of this eugenic idea, but in actuality, this was only the end of coercive eugenics. Moreover, it was the end of race-based eugenics. Scientists have been complaining for a long time that this idea that this was patterned by races was bunk, and the Nazis showed how ridiculous this was, and coercion was no longer a possibility. So these people came to be known as reform eugenicists. But in the early 50s, no one knew what genes actually were. I mean, it was this sort of conceptual idea that 2 people have a baby and there are these elements of heredity, but what exactly was it? Well, here in 1953, we finally find a very young Crick and Watson here who say, here is the actual chemical structure of DNA. So the first time, people began to think, well, if genes are actually chemicals, maybe we can modify the chemicals not by stopping certain people from showing up in the country, not from getting certain people to mate with each other or not mate with each other, but actually through chemically modifying the genome. More importantly, all the techniques of influencing whose sperm meets whose egg have the following limitation. You can only produce babies that those 2 people are capable of producing. You get a tall person, they mate with a tall person, they'll produce a tall person. They're not going to produce someone who's 15 feet tall. There are no humans who are 15 feet tall. But they're thinking to themselves at this time, well, if I could find the gene for height and give someone 10 copies of that, we could give someone who's 15 feet tall. They're more interested not in that, they're interested in intelligence. If I could find the gene for intelligence and give someone 10 copies of that, you could create someone who's, 10 times as smart as everybody else. This was obviously a long ways off. We get to the 1960s here, and with the advent of things like amniocentesis, you could select some very limited qualities of fetuses, selected against things like Down syndrome, but this is not the kind of thing that you're going to be able to modify the species because people don't take abortion lightly, and there's only a few conditions you could really screen for. This starts to change in 1990 with another term I will not use again, pre-implantation genetic diagnosis, which I will call PGD. Okay, so pre-implantation, so this does not require abortion, but it does involve destroying embryos. People generally are more comfortable with destroying 8-celled embryos than aborting a fetus. The way you do this is you start with in vitro fertilization. Okay? The idea the big innovation was, let's say you produce 5 eggs in a dish. Then you let them develop until they're 8 cells, and then you stop that development. You take 1 cell out of each one of them, which amazingly to me to this day doesn't seem to cause any harm, which is pretty astonishing to me. You test it for its genetic qualities, okay? So in a regular Mendelian recessive genetic disorder like Tay-Sachs and things like this, 1/4 of the eggs you produce, the embryos you produce in this way, are going to have the disease. You test them, you discard them. Half of them will be carriers. Well, you're a carrier. That's how you produce in the first place. That's not such a huge deal. 1/4 of them won't have the disease at all. You take those, you implant them in the woman, and you're guaranteed to have a child that doesn't have genetic disease. So this was quite an innovation for people like who had cystic fibrosis and the like. It resulted in an explosion of genetic traits, because the other thing that's happening at this time is people are starting to understand the genome. People originally had identified the location on the genome of just a few of these things, but then there's just this explosion of where these things are. The British government actually regulates this in a very light way, but they do keep track of all these sorts of things. And this is just their list of things you can use this technology for in Britain. This is just AA through AL, and I can't say most of these things, but I'll say a few. Whatever Alagille syndrome is, Alpers syndrome, Alport syndrome, but let's keep a few in our mind here for a few minutes. One is early onset Alzheimer's disease, and the other one I will return to is achondroplasia. So keep those thoughts in your mind. In the US, there's no legal limit on this, unlike Britain. A few years ago, a clinic in Orange County supposedly announced that it would use PGD to screen embryos for eye and hair color, but I think they reversed themselves after public outcry. One thing to keep in mind about this, which is that this does destroy embryos, but it's more palatable to most people than abortion, but it requires IVF, which has its own host of troubles, and you aren't going to do this lightly. You have to superovulate the woman, you have to get these eggs out, you put them in a dish, and then more importantly, once you've produced these embryos, they don't all stick when you put them back in the woman. So this isn't like, you know, a magic bullet, but lots of people are doing this now. So, let me return to my ethical divide here. I've been describing germline modifications of a sort, and that you're picking among embryos, and you're influencing then your children's qualities and those children's qualities and so on. Okay, so I'm here in the germline column. So for cell 2, in this traditional moral divide, normal state of health are genetic diseases. So we can then do cell 2 without engaging in any sort of futuristic chemical CRISPR things, because most people could use PGD to make sure that they do not have children that have Tay-Sachs, cystic fibrosis, and these sorts of things, which took a lot of the energy out from behind calls to engage in gene splicing, genetic editing, and the like. Cell 4 can't be done with PGD, essentially, because the things that you think of as enhancements, let's say intelligence, okay, we could in this room not even agree on what that means, let alone find a gene for it, okay? So that's not at present possible. But moreover, like with PGD, the thing to keep in mind is that if my wife and I produced 1,000 embryos, okay, no matter what happens, none of them are going to be as smart as Einstein. Because my wife and I are not as smart as Einstein. We're not going to produce babies like this, okay? And so therefore, to really modify the human species, you can't pick from among traits that normal people can produce. That's just like, this PGD is sort of like a forced form of mating in a way. So what happened was that this, critically for this current debate, this line here between disease and enhancement started to break down with the question of what is health? So back in the day when we thought of genetic disease as things like Tay-Sachs, it was essentially normalcy. Health was genetic normalcy. Tay-Sachs was not normal and it was bad. The moral advantage here that kept this nice and clean was that nobody disagreed that these diseases were bad. No one says that cystic fibrosis is good. But as knowledge of what genes do increased beyond these single gene recessive disorders, this line blurred in a number of ways. What about genetic deafness or dwarfism? Okay, well, that's not normal, quote unquote, but these people live lives as full lives, right? So recall, achondroplasia is one of the traits in Britain that you can select against with PGD. You can make sure that you do not have a diseased embryo, diseased meaning has achondroplasia. But is that really disease? I'm kind of a sucker for British formality, so I will introduce this guy properly. He is Sir Thomas William Shakespeare, 3rd Baronet, whatever baronet means. But he's also a professor of sociology at the University of East Anglia in England. He has achondroplasia. His father, who was a physician, was the 2nd Baronet, who also had achondroplasia. And he would say that if people want to use PGD in order to guarantee that they have a child with achondroplasia, they should be able to do that. And there have been people who have tried to ensure that their children are deaf using this technology. So in general, these people would question whether or not that's actually a disease. What is a disease starts to become a little less obvious. What about a genetic condition where the child would be totally normal most of their lives and then develop a disease much later, early onset Alzheimer's disease? You can do that in Britain too. Is that a disease? It's at least a different type of disease. What about things that change your odds of getting disease? Some genes increase the odds of getting breast cancer later in life, but does not determine that that will happen. Is that disease? Is that a disease gene? So, despite all this fuzziness, the traditional moral line here has been sort of holding. Most people do not see something as a modification or an enhancement if two people could conceivably produce a baby like this. If two humans somewhere could have come together and done this. But CRISPR, I think, is now about to destroy that moral line. This is George Church. And he's always been one to push back against ethical conventions as hard as he can. He's a very influential biologist at Harvard. In his lab, what they're trying to do, to quote one magazine article, he's trying to, quote, install genes that offer lifelong protection against infection, Alzheimer's, and maybe the effects of aging. Now, there is no human who is genetically immune from aging. What we're talking about here destroys the line because what we're talking about is an enhancement in the name of relieving disease. So this gets us into sort of moral no man's land. People used to sort of be able to perhaps lazily rely upon nature, quote unquote, because we were fixing errors in nature's genome, or you could also rely upon God's design was like this and we're just fixing, completing the creation or fixing the effects of the fall. But what this does, it makes you realize that it's up to us. We have to decide whether or not we want to do this. And this is what the CRISPR creator was talking about with the Hitler reference, which is that what disease is, we have to decide what a disease is. And then she's worried that if Hitler could have decided what a disease was, she says that would have been bad. So, we could heal what we consider to be a disease. What should we do? So let me finally return to gene editing with CRISPR. How does this change this long-running debate? Well, let me go through the cells briefly. Again, cell 1, which is changing genes inside of your body for diseases. There's really no ethical controversy here. The question is whether it works and whether it's safe. Okay? That's what's open for question there. Cell 3 is more morally dubious. Okay? The things that have been mentioned to me by scientists who know what they're talking about is like giving someone their blood a different type of blood that allows them to process oxygen better, and you could win more bicycle races. Okay? You'd be like an enhanced individual human. And that's controversial, but I want to skip it in the name of time. It's not really something as controversial, as cell 2, which is where the ethical action currently is. In fact, the reason that people have called for a moratorium is that people are doing this right now in Britain with embryos. They're not going to put it in a woman yet, but they're trying to see how you would actually do it and let it develop and see what happens, and I think they're trying it in various animals and the like. So, but if you did this, let's say you modified an embryo to remove, sickle cell anemia gene, edit that to make it correct, you would then change the genes of that baby would be healthy, and all their descendants would be healthy as well. So, cell 4 is so controversial. There are many people who want to actually enhance the human species too. The reform eugenicists I told you about never actually went away. They believe in voluntary eugenics, okay, in the name of things like improving people's intelligence. They're called transhumanists. How many people have heard of transhumanists? People of a certain age will get a kick out of this. So basically, the way to put it is that this, what I have here is humanity 1.0. Doesn't work very well, need glasses. Turns out this body's going to fall apart before I get to be 100 years old. Turns out I require lots of sleep, etc. But we could create humanity 2.0. That would be better. We could live to 150. We wouldn't get disease. We wouldn't need glasses, etc., etc. And the question they say is, why not? If we could avoid the inequality concern about two societies and the like, why not? They would say. So, I'll get back to that in a little bit. Let me add some sociological qualifications. This is the one thing I think I add to these debates, which is some of these things might be ethically troubling, but if no one's ever going to do them, I don't think we need to worry about it, okay? So let's try to think what we know about society, what people would actually do if they had their hands on technology. So the first thing to keep in mind is that this technology is not really treating a disease, but rather it's treating the desire of parents to have genetically related children who are free of disease. This has long been noted by critics. And that if Americans were less concerned with having genetically related children, more interested in adoption, and adoption was easier, there'd be a lot less reproductive genetic technologies in general, and you wouldn't need things like CRISPR because people who knew they had these genetic traits would either adopt an actual baby, you can adopt embryos now, you could use a sperm donor, and so on and so forth. So this has always been pointed out, but now we should also say that the parental desire for genetically related children is very strong in the United States, and many people feel very strongly about that. Second is, in terms of likelihood of use, unless you envision all poor people in China, India, and Africa reproducing through IVF, and envision worldwide universal health care provision, this will remain the technology of rich people of the world. Poor countries can't even give their people basic health care. They're not going to engage in this kind of business. And so the vast majority of humans are going to be made what I call the old-fashioned way. How would you actually do this technology? Well, how you would actually do it will affect its use. One would be to modify embryos in petri dishes. So that requires IVF, which is going to limit its use to rich people, requires superovulation of the woman, the embryos don't all take, et cetera, et cetera. But what would make this very popular, easier, would be changing to sperm. Men don't mind having sperm outside their body, but scientists tell me that you can't apply CRISPR to sperm because the genetic material is too tightly crunched up in there, so you can't do that. You could use CRISPR to modify the stem cells that produce sperm. So inside male testicles, there are cells that make the sperm continuously. If you modify those, you would modify the genes of the man's sperm, which would then hit the egg, produce a genetically modified baby, and you're off to the races. Now, I think a little bit of knowledge of human experience is useful here, which is that the way you would do this is you would either use a knife to remove tissue from men's testicles, change it, and put it back, or at best, insert needles in men's testicles. And I think we can assume that this is unlikely to be popular without major improvements and will only be done by very highly motivated people. The path to the brave new world may be saved by male squeamishness. So, the other thing is you don't need a PhD to know that people will act in such a way as to bestow advantages to their children and their families. Obviously, this is what happens in America in all sorts of non-genetic ways. You know, people will sacrifice and save every last penny of their entire lives to send their kids to Harvard. It's just the way that that works. So, what's the ethical debate? We have gone over the ways that people have had to influence the genes of their children, the broader species, and the sociological limitations on gene editing. So before I get to the debates themselves, let me talk about what there's absolute ethical consensus about among everybody, which is none of this should be done if it cannot be shown to be safe for everybody involved. This is the concern that these scientists who know about these risks are telling us about. Would you introduce some damaging genetic sequence into the entire human species that can't be recalled? How would you recall this mistake? I think the only way you would do it is you'd have to find the descendants and make sure they don't reproduce. I would guess. Okay? Someone talked about putting engineering in a kill switch. I mean, I think, they want to make sure that they understand what this would do first. Okay? But morally, everyone needs to keep in mind, safety is just a precondition. Okay? Just because it's safe doesn't mean it should be done. So, this is the type of argument, this is the structure of most of the arguments that you need to understand. This is a slippery slope argument. There's good slippery slope arguments and bad slippery slope arguments. The way this works is you start at the top of the slope on something that everyone agrees with would be fabulous, okay? And that leads to something in the middle at time 2, which is something you wouldn't want to have done at time 1, and leads to the bottom at time 3, something you really didn't want to have done at you started, okay? This takes time. The typical argument is that 10 years from now, you'll be at time 2. 10 years after that, you'll be at time 3. There's good and bad slippery slope arguments. The bad ones are the ones that claim inevitability, that it is inevitable that when you step out of the slope in a number of years, you are inevitably going to be at the bottom. Those are considered to be rhetorical, and the reason is that they assume that people in the intervening time have no decision-making authority and don't do anything. It just sort of happens. The good slippery slope arguments are probabilistic. They don't give you certitudes, but they sort of make educated guesses about what society is like and how things will or not change as these things develop. So here, an example would be, imagine that California approves a law to allow assisted suicide for people who are terminally ill, okay? That's step 1, okay? The slippery slope argument might say, well, in 10 years, because people get used to the idea of people committing suicide in this way, people will say, well, why are you just doing it for that? Why don't you do it for something that's not quite as severe as terminal illness? How about people who are suffering in some sort of other way? And then once people get used to that, 10 years later, before you know it, we will be euthanizing people who are not useful to society. Well, that argument only works if you can explain the sociological mechanism as you go here. To have a slippery slope argument, you have to say you don't want to be here at the bottom, right? That's what you're trying to avoid. So what's the bottom that people are trying to avoid? People are trying to avoid cell 4. People in general are trying to avoid situations where we have genetic enhancements for things like intelligence. Okay, there's some exceptions. Some people welcome the bottom of the slope, and so they don't really care about the slippery slope. They say, let's get there faster. But most people want to avoid that, and they want to avoid a situation where you have a society with two essentially subspecies, the genetic haves and the genetic have-nots. The bottom of the slope here is what ethicists will call Gattaca, okay? So some of the youngins in the crowd up there might recognize that reference. How many of you seen the movie? Whoa. There we go. Wrong one. Gattaca. This is a movie that tells a story where everything in your life happens due to your genes, your occupation, who you date, who you marry, where you live. It's all determined by your genetic qualities. Here's the trailer for the movie here. You know, a single drop of blood will test and tell you exactly what will happen with your life. Ethan Hawke is the genetic inferior, as indicated by the fact that he's wearing glasses, which means he was not designed properly. He is slated to be a janitor the rest of his life because of his genetic sequence, but he dreams about being an astronaut. Being an astronaut is limited to the genetic superior population. And of course, it wouldn't be a good movie without a good love interest, which is Uma Thurman, who plays one of the genetically better people who cannot date Ethan Hawke until she tests his genetic sequence. She takes a piece of his hair, she takes it to the lab, she tests him. You get the idea. See, this is a society what we would call durable inequality. It's not just inequality that's like over like you can inherit the money from your parents. It's written into your body itself, and this determines what happens in your life. So this is what many people are trying to avoid, and this idea of modifying for intelligence. So I think this is the best description of the debate as it currently stands. Yes, there are arguments that don't fit into this structure, but this is basically it, which is most people want to avoid Gattaca here at the bottom. Most people don't want people to suffer from diseases like sickle cell anemia and Huntington's disease and on and on and on. So if you start at the top, are the odds high enough that after however many years, you are going to be at the bottom? Okay? So I should add at present, 15 of the 22 countries in Western Europe have decided not to get on the slope at all. They have laws, treaties, policies that ban any germline modification at all for disease or anything else. It's basically correlated with the proximity to the Nazi experience, actually. Indeed, Germany refused to sign the convention that did this because they thought it wasn't strong enough. But there's no laws like this in the United States. I mean, you can't just go genetically engineer someone. There's all sorts, you have to get past the Food and Drug Administration. It's not easy, but there's no specific laws about this. So let's start with these arguments for people who want to step out onto the slope here and use CRISPR to treat Mendelian diseases like sickle cell anemia. So again, if you were paying attention, you'd say, why would people who are carriers need to do this when they can use PGD anyway? It's just so much simpler, okay? Well, it turns out that there are people, for technical reasons, who have these diseases who can only produce embryos that have the disease. There's apparently 40 couples in the UK who want to use PGD to avoid serious genetic disease but can't. And if I've not had anyone be able to explain a number to me for the US, but just extrapolating from the population differences, let's say there's 200 couples in the United States who can't use PGD to produce genetically related children. But even for the larger group who are carriers but could use PGD, PGD is difficult, imprecise, destroys embryos. Some people cannot produce enough embryos to effectively select from. If the technology could advance to where men's sperm could be changed, these people could reproduce more easily. The suffering that prospective parents feel from not being able to have genetically related children is real and needs to be acknowledged. The existence of the entire infertility industry testifies to how strongly people feel about this. The adoption of babies is difficult and expensive in the United States. While using other people's eggs or sperm would be easier, that involves in vitro fertilization, which doesn't always work, as I mentioned before. And finally, in the US, there's a strong tradition of individual rights, in particular regarding reproduction. And it has been argued by many that the government cannot say what the qualities of your children can or cannot be. You have reproductive liberty. So people who make this argument then argue against the slippery slope by saying that all these counter-arguments using the slippery slope are vague speculations about cultural harms in the future that we can't really quantify that are going to be compared to people in my doctor's office today. So, what are the arguments against? The ethics debates among people who are called bioethicists and scientists typically want the debate to be limited to between individuals. There's some people in the present who want to have babies, and to do the argument, you've got to find someone in the future who will definitely be harmed, okay? And if you're constrained to make arguments like that, really the only argument you can make is this one, which is that people in the future who do not yet exist haven't given their permission to be experimented upon. Now, I consider this to be a straw figure in that we don't ask people in the future anything. Like, we're currently destroying the planet for them, and we didn't ask them permission. We might end up saving the planet. We didn't ask them about that either. That's just not how society works, okay? The more serious argument against are these slippery slope arguments. I really do think that if you took one of the people who's opposed to all germline engineering and said to them, really, are you really opposed to germline engineering for Tay-Sachs? They would probably say, no, I'm actually not, if you can guarantee me that it doesn't continue on the next step down the slope. People are really concerned about ending up down in Gattaca. So the argument is that the risks of the slippery slope are just too strong. Once we got really good at changing stem cells to produce sperm to avoid sickle cell anemia, there would be social pressure to move on to these fuzzy diseases, propensities to disorders, and deafness and the like. And then after that, it would be, well, why not do these sort of enhancements, et cetera? Your assessment of that argument depends on how you think American society actually works. Once we got good at that, there would be an entire industry of lobbyists and everything to back up their views, and they would use the same technology to make disease enhancements and the like, make your children less susceptible to disease. And again, if you think that's how American society would work in the future, that would be a greater concern for you. And I should add that the fact that I'm having this conversation with you today indicates the legitimacy of the slippery slope argument, in that basically on the slope, people consider these things to be like little walls. Like, can you imagine a wall that will stop? The germline was supposed to be the wall. For decades, that was the wall that no one was going to go past, and that was going to stop people from getting to Gattaca. But now that it's actually possible, all of a sudden, now we're talking about skipping through that wall. And so I think this is how sociologically these debates actually occur. So the people who say we're worried about the slippery slope say that people's reproductive rights are not absolute, and that such people who would have a genetic disease but can't use IVF, so there are these few people that they should adopt. This is essentially what their argument is. Now, believe it or not, that's the simple debate. Now let's go on to the challenging one. Some scientists are already a few feet down the slippery slope. With George Church's goal, for example, which is to make us resistant to disease. This is far off into the future, but people are already arguing about we need to set the ethical standards in a particular way now so that this would be morally acceptable if we figure out how to actually do it. This is not returning someone to normal, like stopping someone from having Tay-Sachs. No human is resistant to aging, okay? It's just would create a new type of human. So what's the argument for doing this? Well, obviously, let's be clear. The reduction of suffering, okay? Think of the suffering you would eliminate. If people didn't get cancer, no longer debilitated by infection, genetically immune to HIV/AIDS, did not get heart disease, okay? So, let's be real. This is quite a moral good that we have potentially hanging in front of us. And I'm not going to dwell any more on this because I think everyone understands this and how powerful this argument is. But who would argue against it? The rest of the arguments I'll talk about in my few remaining minutes against editing are not only made for this particular case of editing, but for anything further down the slope. The first is it's often expressed as a religious argument. It also has a secular form. It requires a certain religious sensibility, which is to say it's part of being human to modify nature and to heal the brokenness of humanity, but for humans to modify the essence of humans themselves is beyond our role. The reason this requires a religious sensibility is you have to have some sense of what humans' role actually is, okay? But that's a typical argument. people don't, as in a sort of aside, people don't realize this, but the largest religious groups in the U.S., such as Catholicism, evangelical Protestantism, and mainline Protestantism, would not be opposed to germline modification to return to normalcy, provided certain conditions are met. They would say, we should play God as God plays God. That is, playing God is fine as long as you're doing what God would want you to do in the first place. They would consider this to be part of the mandate. Christians would say this is part of the mandate to heal the negative effects of the fall, which is what we do with medicine in general. Judaism has a similar idea called tikkun olam, to repair the world. Most of the Christian groups, at least, would stop at modifying humans beyond returning to sort of normalcy. Okay? Again, to use sort of more theological terms, the term they would say is that we are co-creators with God, or created co-creators with God, but we're not God. So that's the basic divide, they would say. And further, I should say that the additional conditions that some of these religious groups would probably not be met. Evangelicals and Catholics would say, the official churches at least would say, that you can't destroy any embryos in doing this, which is probably impossible. Catholics would go further and say you can't do this outside of the Sex Act. But I'm not a Catholic theologian, but from my reading of it, the modify the stem cells in the testicles version, and then people could have sex and have babies the old-fashioned way would work. But the main argument, I think why most religious groups would not really end up going for this, is that this religious sensibility is very attuned to slippery slope arguments and social effects and social harms. And so I think they probably wouldn't agree in the first place. Second argument against is this would result in two populations of humans. Essentially, yeah, there's all sorts of inequality now in America, and this would just make it worse. In principle, poor people can currently become rich. I mean, my colleagues who study this kind of thing will tell you that, yeah, it's gotten a lot harder. Intergenerational mobility of wealth has gotten a lot harder, but it's at least still in principle possible. But the previous debate I was telling you about, producing children that do not have genetic diseases, doesn't result in durable inequality. Modifying your children to have them not have Tay-Sachs doesn't give them any advantage. Okay? It just makes them normal, like everybody else. But if you created a child that was genetically resistant to aging, this would be a benefit. Moreover, from what we know of who's going to do this, these would be the children of very wealthy people. Okay? So we know that people go to great lengths to give children advantages in life. Tennis lessons, piano lessons, private schools. I don't think it'd be hard to imagine. This first technology is not going to happen in the US. It's going to happen in the UK, China, Singapore, somewhere like this. So a couple goes off to Singapore to produce such a child. We'll call that child Adam. Another couple goes there, produces a girl named Eve. You see where I'm going here? Then what sociologists call marital homogamy kicks in. Marital homogamy means who marries who. Do people from the same race tend to marry each other? Yeah. It's not that strong, actually. Do people from the same religions tend to marry each other? Yeah. But what's the new champion of marital homogamy in the past 40 years? Education and class. No one with a PhD is married to someone with an 8th grade education anymore. At one time there was, but no longer. Rich people marry rich people, upper middle class professionals marry other upper middle class professionals. There's entire mechanisms that exist to perpetuate this called elite higher education. You know, you're all familiar with this, okay? So, what would Adam and Eve 2.0 be told? Would they say, go marry whoever you like? You know, someone you think is really interesting. No, they would say, look, we spent all this time and effort modifying you, okay? You better not waste it by marrying one of these, you know, genetic untouchables. You have to stick within people like this. And before you know it, you know, you don't need to modify 100,000 people using CRISPR. You just need probably not 2 because of interbreeding problems, but let's say 4 or whatever, you know. And all of a sudden, you have 4 people who have had testicular surgery, and you've got these modified people. So, you know, this idea of durable inequality taking off, you know, again, assuming that they can figure out how all this works, but they seem to be pretty confident, is a big concern to people. And then the final 2 concerns are what are called cultural damage arguments. these all assume that technology is not neutral, okay? You might say technology is neutral, it's just how you use it. But things like nuclear weapons, you can't bake a cake with a nuclear weapon. They only have limited purposes. Your choice, the technology itself structures the moral choices you have. The same thing is that these technologies teach us something about humans, okay? And the first of the argument is that if we start creating children who are designed by their parents, we will think of them ever so slightly as the same as other things that we design, which we consider to be objects, okay? Of course we design our children all the time by trying to make them take piano lessons and the like, but the argument is that because the design is literally built into their bodies, it's stronger and more durable because the children can't say no, like they can eventually to the piano lessons. There's the secular version of this argument, the religious version. The secular version is most associated with Harvard political theorist Michael Sandel. He would say that we do not want to control every last aspect of the lives of our children, but have, quote unquote, openness to the unbidden. Genetic design of our children distorts the relationship of parents and children. We would be less likely to accept children as they are, but instead see any of their failings as failings of our own design efforts. His theory is that if you design them so precisely to perform in some way and then they fall short, you will think of them as having failed in a more fundamental way. So he thinks that seeking perfection in children makes it more difficult to engage in unconditional love. And we will come to think of our children not as what he calls gifts, but as objects. And therefore, if they think of them as objects, we will in an ever so small way treat them like objects. This isn't hypothesized to be noticeable, a big enough effect to be noticeable within one generation, but the theory goes, after 15 generations of this, we would, if we could somehow measure the difference, we would think of people slightly more object-like and therefore treat them more object-like. A similar claim, Christian theologians, building on a piece of the Nicene Creed, Gil Meilander describes the Son of the Father as begotten and not made. So like good theology that enters the public sphere, he translates it into secular language. Meilander thinks that children should be begotten and not made. Like Sandel, he's worried that if we design our child, we're making him or her. He would rather have us not design them, but to be open to the mystery of a child we do not control completely. The basic insight here is that you're not equal to something that you make. Anything you make, you are by definition the master of. So I didn't make this thing, and it didn't work apparently very well. But let's say I made it. I'm the master of this object, okay? The idea being if you truly make your child, you will come to think of that child as something you are the master of instead of eventually a co-equal with yourself. I mean, I've got kids. I do not think of I guess I'm being recorded here. But anyway, I don't think of my teenage kid as equal to me, but eventually I will. Meilander writes that when the sexual act becomes only a personal project, so does the child. No longer then is the bearing and rearing of children thought of a task we should take up as a return we make for the gift of life. Instead, it is a project we undertake if it promises to meet our needs and desires. Now again, the problem with this debate is that if you're looking for quantifiable evidence to weigh against people in the doctor's office right now who want to have genetically related children, you're not going to get it with these arguments. These are supposed tiny effects that aren't going to kick in for some 40 years of doing technology, and therefore it's a sort of a mismatch. It sort of depends on what you think a legitimate ethical argument is. I tend to think that these sort of more vague arguments that are often expressed actually in literature and movies and more sort of speculative narrative things are something that are important that we should remember in these sorts of debates. But that doesn't answer the question of whether or not they're correct. So, there we have it. Should we go forward with the modification of embryos to allow people to have genetically related children while remaining free from disease? Should we modify the children to make people live longer, be smarter, and so on? And I know you've been waiting the entire time to give you the actual answer. And unfortunately, it's up to you. Don't defer to me or some other expert. Look to scientists to tell you what's actually possible. It's very informative to know, like some of these ideas, like we can wipe out some gene from the population, and then you listen to the scientists tell you that's actually not possible. Mutations happen faster than that. Some of these genes are actually being carried for some of these things is good for you. Being a carrier for sickle cell anemia confers resistance to malaria. That's actually good for you. You just don't want people to have the disease. So listen to scientists, but then decide morally what you think, and everyone's view of this will differ depending on what your values are. I've tried to set your choices here in a little bit of structure to make it a little easier for you. I hope you found it useful, and I look forward to answering any questions you have. Thank you very much.